In this lecture, we'll talk about developing transgenic plants expressing insecticidal toxins of Bacillus thuringiensis, basically to develop insect-resistant plants. So I'll give a brief background about the bacteria Bacillus thuringiensis. Ishiwatari in 1901 isolated the Bacillus thuringiensis strain from Bombyx mori. BT Bacillus thuringiensis is abbreviated as BT. It's a gram-positive facultative aerobic rod-like motile and sporulating bacteria. So it forms sporulation during adverse environmental conditions. BT is naturally occurring soil-borne bacterium that is found worldwide. Ubiquitous in nature, it is present in all types of environment. Produces crystals of endotoxin, or also called cryprotein or delta toxin, and which is found to be toxic to insect mainly during their larval stage. And hence these toxins can be used as a insecticide. These crystal proteins are insect stomach poisons. Insects stop feeding within 2 hours of first bite. So once they ingest this toxin, they stop feeding within 2 hours. If enough toxin is eaten, then larvae die within 2 or 3 days time. So these are considered as important biological agent for controlling insects. BT crystals, sometimes referred as insecticidal crystal protein or ICP, are protein crystals formed during sporulation in some BT strains and is coded by the cry genes. So the genes which encode these proteins are called cry genes. So this is some more important milestone in the development of BT as a biopesticide. In 1901, Ishiwatari first isolated the bacterium Bacillus thuringiensis as the cause of the Soto disease in Bombyx mori. In 1915, Berlin reported the existence of a crystal within BT, but the activity of this crystal was not discovered until much later. In 1956, researcher Hannah James and Angus found that the main insecticidal activity against Leptopteran insect was due to the parasporal crystal. So this was one of the major discovery, major milestone you can say in field of Bacillus thuringiensis. In 1958, in the US, BT was used commercially. So once it was known that these toxins are very effective in killing insect larvae. So this was in 1958 in US, particularly farmers started using this BT commercially for controlling insect pest. In 1961, BT was registered as pesticide to the EPA. In 1996, BT cotton was introduced into US agriculture. So this 1996, another major milestone, transgenic cotton was developed, which was the expression insecticidal crystal protein of the BT, and it was introduced in the agriculture for controlling insect pest. So the variety of strains have been isolated. Now there are several strains are available now. A variety of strains have been isolated from different habitats, and today more than 100 crystal protein genes have been sequenced. So this is the electron micrograph of the one of the crystal protein of Bacillus thuringiensis. You can see the spores here as BT is a spore forming bacteria. So you can see the spore here, and the bipyramidal shape. You can see the crystals here. So these are the crystal protein, insecticidal crystal protein of the BT. This slide shows the 3D structure of the crystal of Cry2A, one of the toxin. This structure is represented with 3-domain toxin produced by BT. Roman numeral indicates the typical domain of the 3-domain Cry protein. It has 3-domain 1, 2, and 3 domain. First domain is perforating domain, form the pores and all that one. Central domain involved in toxin receptor interaction. The second, this particular domain is involved in binding to the receptors in the insect gut. So this is the one which binds to the insect gut. And third is galactose binding domain involved in receptor binding and pore formation. So this part, domain 3, is involved in both pore formation as well as receptor binding. So this is the 3D structure of crystal protein of Bacillus thuringiensis. So as you know, there are several strains of Bacillus thuringiensis, and they produce different types of crystal protein, and each protein have a specific target. Like this strain Berliner, The protein size is 130 to 140 kilodalton. It is the specific. It kills specifically. It kills leptospirosis strains. Similarly, Kursaki SD1 also 130 to 140. It also kills leptospirosis pest. And the Kusaki SD1, another which produces around 71 kilodalton protein, it kills leptospirosis and dipterin both. Tenebrionis, this is another unique strain, Tenebrionis, which produces around 66 to 73 kilodalton protein. It kills beetles, coleopteran species. Then we have another important strain, Israelensis, BT Bacillus thuringiensis subspecies Israelensis. It was discovered in Israel, so that's why we call it Israelensis. It produces a crystal protein of 68 kilodalton, and it kills dipteran, that is mosquitoes and all that one. Okay, so there are several strains of Bacillus thuringiensis are available, and all these they produce different types of protein, different sizes of protein, different types of protein. Some are bipyramidal, some are cuboidal, some are different shapes they have, some are oval, and each protein has a specific target. There are target insect for BT toxin. Crytoxin have different activities against insect species of the order Leptospirosis, particularly moths and butterflies, because this is the main which harms the group from the insect from this group. Leptospirosis are the major pest of our crops and all that one. So these strains which target this are very important. Dipteran flies and mosquitoes. As you know, mosquitoes carry is important vector for carrying so many diseases to humans and all that one. So these mosquitoes also can be controlled by Bacillus thuringiensis, particularly from Israel subspecies Israelensis. Coleopteran beetles. There are many crops are damaged by beetles. Also, there are pests, soil pests, which belongs to beetle family. So and some other targets are hymenoptera like wasps, bees, ants, and sawflies also. So there is basically this slide just to show you that BT toxin can target different types of pests, and they are highly specific also. Just like Israelensis strain will kill only mosquitoes, they won't kill any like moth or butterflies. Okay, so they're highly specific protein. So this shows the whole spectrum of BT endotoxin, like delta endotoxin. Major target is leptospirosis pest, and there are several genes are there. Cry1a, Cry2a. You can see so many different genes are there which cause protein of different sizes and all that one, and they target leptospirosis pest. Similarly, some toxins like Israelensis can target mosquitoes, dipteran. Some toxins from different strains can target coleopteran like beetles, and it has been some can even some strains have been found which can kill cancer cells also. Okay, so they have different genes. Similarly, gastropoda, snails, hymenoptera, hemiptera, or even rhabdite, or some strains which can kill even the nematodes also. So they have nematocidal activity also. So this is the whole spectrum. So you can, from this slide, you can understand that this BT strain is, this bacteria particularly is so important for the mankind. So in this now we'll try to understand how the BT works, how it kills the insect pest. The first step is that this toxin this is a protein, and this toxin has to be ingested. So particularly the larval, only the main target is the larval stage of the insects. So larvae should ingest this one. So when it gets ingested, it gets solubilized, and then it gets proteolytically activated. Okay, in the insect gut, and once it gets proteolytically activated, it binds to the target receptor sites in the insect gut, and then it forms the pores and toxin lesion, and then it will kill the insect larvae. So this is how it works. So you can see in this picture. So BT toxin, as you can there are two ways you can spray BT toxin. One is directly you can take spore crystal mixture from the BT strains, isolated from BT strains, and then spray on the field. Or the second one is that you can introduce these BT crystal protein genes into plants like BT corn, BT cotton. So this is corn expressing BT toxin genes. Similarly, BT cotton means cotton plants expressing BT toxins. So once insect larvae feed on this one, so like BT crystal toxin will get ingested. Okay, it will get solubilized. Because it's a crystal, it's inclusion body type, so it gets solubilized in the insect gut, and once it gets solubilized, it get cleaved and it becomes proteolytically activated, and once it get cleaved, it get activated, then it binds to the receptor. Here you can see the toxin monomer. It has like the particular domain, which is domain 2 and 3, which are involved in binding to the receptor. They will bind to the insect midgut cells here. So this binding will occur. Once this binding occurs, they start forming the pores. Like here you can see here the pores will be formed in the insect. Okay, the pores will be formed, and once this pores are formed, it will lead to the osmotic cell lysis. When the osmotic cell lysis occur, it will lead to the cell death, and then septicemia, and the larvae will then finally die. So this is the mechanism of action of BT crystal protein toxin. So this is how actually this just shows the activation, solubilization activation of this one. So crystal protein is dissolved, activation, and it become activated to form toxin. So this is basically earlier this one, when this is the one crystal protein which is ingested by the larvae. So this is protoxin basically. It get with the help of the proteases in the insect midgut, it gets cleaved, and active toxin will be released then, and this active toxin will then go and bind to the receptor in the midgut. Okay, once this binds to it, form the pores in the membrane, gut membrane, and then once pores are formed, then it will kill the larvae. Now we'll learn about the application of delta endotoxin or the insecticidal crystal protein of Bacillus thuringiensis. Important role you can see is we can develop these protein as a bioinsecticides. Insects are the most abundant group of organisms on earth, and they negatively infect the humans in variety of ways. They cause massive crop damage. So there are two ways they harm human beings. They cause massive crop damage, okay, cause a lot of and they act as vector of both human and animal diseases. So both of these can then be controlled by using BT as a biopesticides. So you can see that damage. So they can damage the shoot, stem, leaves, or the fruit also. You can see the fruit damage also. They can damage fruits also. So this causes extensive loss to the crop, and as well as economic loss to the farmers. So basically this is the life cycle of fruit and shoot borer, and you can see the life. So this is the egg. From egg they become larvae, then pupal form, and the adult form. So larvae of the insects are the one which feeds extensively on the crop. Okay, so they are the one which causes extensive damage to the crop, and BT toxin generally target larvae only. Okay, so this stage BT toxin targets insects. So before we go to the biopesticide, we'll know some of the why do we need biopesticide. So before we'll go through some of the chemical pesticide drawbacks of the chemical pesticide. As you know, most of the time farmers are using chemical pesticides, and which is causing extensive damage to our ecosystem. So during the 1940s, a number of chemical insecticides were developed for controlling insect population. One of these was the chlorinated hydrocarbon, that is DDT, dichloro-diphenyl trichloroethane. DDT was very effective. It was found when it was discovered in 1940, it was found very effective in killing and controlling many species of insect pests by attacking the nervous system and the muscle tissue of the insect. So this was the mechanism of its action. It target nervous system and muscles. Other chlorinated hydrocarbons such as dialdrin, aldrin, chlordane, lindane, and toxophene have since been synthesized and applied on a massive scale. By 1996, over 100 million acres of US agriculture land were being treated annually with the chemical insecticide. However, at later time, researchers realized that chlorinated hydrocarbon insecticide and organophosphate insecticide had dramatic effect on animal ecosystem and human also. Okay, so it was found out in 1960s only. It was found out after 20 years of extensive usage. It was found that it is causing harm to our ecosystem as well as to human health also. DDT is found to another important drawback with using DDT and other chemical pesticides, that DDT was to persist in environment for 15 to 20 years. Basically, it was not easily degradable. So 15 to 20 years it was able to persist in the environment and accumulate in increasing concentration through food chain. So food chain it enters the our body then. Okay, so keep accumulating increasing through the food chain. This bioaccumulation in fatty tissue was having a significant biological impact on many organisms. For example, in North America, many species of birds, including peregrine falcons, sparrowhawks, bald eagles, brown pelicans, and double-crested cormorants, were severely depopulated. So their population was decreasing because of the accumulation of DDT in the food chain. And later on also it was found that targeted insect pest population, they are becoming increasingly resistant to treatment with many chemical insecticides. So these were the major drawback the chemicals has. So this led to this necessitated the development of alternates, and the biopesticide, particularly the BT, is a major milestone. So this, because of so many drawbacks of the chemical pesticide, researchers started developing crystal protein as a biopesticide. The major attraction of this crystal protein is that the toxicity of these crystal protein against certain insect and their high specificity led to the development of bioinsecticide. As I told earlier, so these toxins are highly specific. If one toxin kills Lepidopteran, it won't kill Dipteran or other insects. So basically it is safe not only for other insects, it is safe for other animals as well as human also. So this is a major advantage of crystal protein as compared to the chemical pesticide. Chemical pesticides are non-specific, and here we have a highly specific biopesticide. This is just an example how it works actually. You can see in this figure, in this one, this is the BT toxin present in the peanuts in the bottom one, protected from extensive damage caused by the cornstalk borer larvae. So you can see in this plate, larvae has damaged these leaves quite a lot. You can see extensive damage. So it was larvae have damaged these leaves quite extensively, and here BT toxin is sprayed on these leaves, and you can see the dead larvae, and damage is very less. Okay, so this is just to show that how BT toxin works, how it protects the plant from the insect attack. But to use BT toxin, there is one major concern about BT toxin directly when it is applied on the field actually. It was found out that crystal protein of BT is very sensitive to UV, which leads to rapid degradation of protein and reduced efficacy. So what if we directly spray BT toxin on the plants, and it is when it gets exposed to UV or sunlight, it gets degraded very fast, and so and then its efficiency is reduced dramatically. So basically it won't be able to protect the protect the plant from the insect damage because of the degradation of the BT toxin due to the UV or exposure to the sunlight. So now that's why scientists designed another approach, that was to transfer BT toxin gene directly into the crop plants. So once it introduced the toxin genes are introduced into crop plants will then produce these toxins which will be inside the plant tissues, and they will be protected from the UV light. So this strategy was developed. So main step for developing insect resistant, so these are called insect resistant transgenic crops. So main step for developing transgenic crop identification of active genes, like here we can say crystal protein genes of Bacillus thuringiensis. Then we transfer the genes to the plants, regeneration for protoplast, callus or tissue. Okay, whether we can use protoplast or callus. If you are using agrobacterium, we can directly use from tissues and callus. Gene expression to the desired level, so we screen for the plant which express maximum level of the crystal protein. Backcross to produce the varieties. Field test. Then you have to do the field test. Once you do the field test and you find these plants are field test to see the efficacy of the crystal protein to protect plants from the insect damage, and once it is successful, field trials are successful, it is approved for the commercialization. So this is how this is method of the gene transfer. So most common method is using agrobacterium mediated gene transfer or bombardment of cells with the particle coated with the genes of interest. But the most commonly used method is agrobacterium mediated gene transfer. So here this picture shows the two different routes for the gene transfer. Here in this you can see the agrobacterium mediated gene transfer. You take leaf samples and then transfer the insecticidal crystal protein genes from agrobacterium to the leaves directly here, and you get the plant. Another one is the particle bombardment method. Okay, so both method can be used effectively to generate the transgenic plants. So this is how the BT plants are developed. Inoculation here basically co-cultivation with the two different agrobacterium which is having TI plasmid as well as the binary vector expressing the crystal protein gene, basically having the crystal protein genes, and then selection based on the kanamycin resistant callus. You select the callus and then start regenerating. So this is the recombinant callus with under the selection media, kanamycin selection media. So the recombinant one which has whatever callus has got the insecticidal crystal protein genes along with the kanamycin resistant genes, so they will able to grow in the selection media. So this callus then we regenerate the whole plant. So once we regenerate these plants, you can do the molecular verification of the gene presence and the expression. One can do PCR to check whether the gene is there inside in the plants tissues, or one can do Western blotting to see the expression of the insecticidal crystal protein genes. And once these plants are ready, they can be tested in the field trials for the insect protection. So BT cotton is one of the important one. First insect. BT cotton was the first insect resistant plant. So this is basically BT cotton is a genetically modified variety of cotton that produces an insecticide, and it was developed and marketed by Monsanto. It's a US company. So once it was developed, India also procured the seeds for this particular BT cotton. So as you know, India is the largest cotton producer and consumer country after China. In 2002, BT cotton was introduced in India. India has the largest hectare of cotton and accounts for approximately 1/3 of the total cotton are planted in the world. So advantage of the BT cotton, it has been found that yield was much superior as compared to the normal cotton plant, because the damage by the insect was very low in this case. So that's why we got more yield was higher yield in this case, in case of BT cotton. Since the yield was high, there was more profit for the farmers, and also lesser need of pesticide. Pesticide was not required, or minimal amount of pesticide. Better quality of cotton because the damage was less, and suitability for the early sowing. It can be used for early sowing also. So these are some of the advantages of BT cotton. So there was a lot of advantage also for using BT cotton in India. For 11th year, BT cotton was planted in India in 10.8 million hectares. Decline in insecticide use was from US $160 million in 2004 to US $25 million in 2010, and 85%. So this was the economical advantage of using BT cotton. So you can see here by the numbers actually in 2004, when 2002 India started developing growing BT cotton in India. So since 2004 In 2004, insecticide worth $160 million was used, but it got reduced to just $25 million in 2010. You can see a dramatic decrease in the consumption of pesticide, chemical pesticide in India. So basically, it became cheaper for farmers to grow these BT cotton in India because they were spending less on the chemical pesticide. And cotton yield increased from 308 kg per hectare in 2001-2002 to 500 kilogram hectare in 2011 to 12. So you can see the increase in, there was a decrease in use of chemical pesticides. It saved a lot of money for the farmers, and at the same time the yield of the cotton was increased from 308 kilogram per hectare to 500 kilogram hectare in 2011-12. So this is just to show you that using transgenic cotton plant, insect resistant transgenic plants, X1 BT toxin is best beneficial for the farmers. So similarly, beside BT cotton, so BT cotton is the only transgenic plant which is approved in India, but there other plants have been developed. You can see here BT brinjal also. 60% of the plant protection cost for controlling fruits and fruit borer is reduced. Once you use BT brinjal, around 60% of plant protection cost for controlling fruit and fruit borer is reduced. So fruit and fruit borer is the main pest for the brinjal. In spite of extensive use chemical pesticide, larvae lead a concealed life. It is difficult to control through insecticide spray. So it's very difficult pest actually, fruit and fruit borer. So here BT toxin plays very important role. Here you can see difference. Non-BT, you can see there is extensive damage to the brinjal here. Here similarly you can see non-BT, there is a lot of damage here, and similarly here you can see the damage caused by the shoot and fruit borers here. But here, where in the case of BT brinjal, you can see the quality of the vegetable here, brinjal, you can see. So there is no damage at all. So this is just to show you that these transgenic plants are highly effective in controlling insect pest. Similarly, you can see BT corn has been developed. So this corn plant explains BT toxin genes. So this is basically this BT corn protect plants from the corn borer. So before this, BT corn was developed. 40 million tons of corn never reaches the market due to the damage by the corn borers. If corn borers are not controlled, the larvae will eventually turn into moss and the cycle continue, and they will keep on damaging the crop. So BT corn produces a protein that gives BT corn built-in resistance to the corn borer, offering protection throughout the plant where the insect attacks. You can see here, this is non-BT. You can see the extensive damage, and you can see here BT corn here. So there is no damage at all. Similarly, you can see here extensive damage by the corn borer. So this is just to show you that if we can use BT corn, we can reduce lot of crop damage also, and it will benefit from farmers with more economic returns. Since 1996, since the development of transgenic plants, there is a rapid increase in development of these plants, transgenic plants, and also these are adoption of this particular technology in the USA also. You can see so many plants have been developed, ST corn, BT cotton, ST cotton, and they are being accepted by the consumers in USA, particularly in the country like USA. Most of these BT transgenic plants have been consumed by the consumers till 2000. This is data is 2005, but still you can see in 2025 so many transgenic plants are available now, and they are being used by the consumers in USA and other developed countries. But there, however, with each technology there are some disadvantages. The main one disadvantage is higher cost of the seeds. Higher fertilizer and irrigation costs will be there, higher harvest costs. So these are some of the drawbacks of using transgenic, but this can be overcome. So there is a question: BT corn effect beneficial insects and natural enemies. Many studies have shown that BT cryotoxin are highly selective in killing larvae of moths. As I told, these toxins are highly selective. BT corn, however, doesn't affect beneficial insects including honeybees, lady beetles, green lace larvae, spider, pirate bugs, or parasitic wasps. So it doesn't kill it doesn't affect basically other beneficial insects. Indirect effects on natural animals, European corn borer, however, could occur. So this is indirect effect. Predator parasite pathogens of the corn borer might decline as corn borer population declines. So this is indirect effect basically. But overall, these toxins are highly safe and they don't harm the beneficial insects. How about in case of human being? So BT toxin has no toxic impacts on higher animals including human beings. The major reason is that we have acidic stomach. We have a very low pH. The low pH is around 1.5 in human. And this toxin requires alkaline pH for the activation, which is there in the insect gut. Similarly, humans, we don't have required receptor for the toxin. So basically we don't have receptors for binding of these insecticidal crystal proteins. So it means that these toxins will cause no harm to the human beings, and a lot of studies have been done now to study the safety of these toxins in human and other animals. So BT is safe to non-target organism including human. So it means just these BT cotton or BT brinjal, BT corn, they can be easily consumed by the humans, and it is being consumed in US and other developed countries.